Hi everybody, and thank you for choosing this video. I will assume that before this video you have watched the other educational lectures in this session about magnetic resonance spectroscopy and diffusion weighted magnetic resonance spectroscopy. So here I will focus mostly on how to tune your diffusion weighted spectroscopy experiments to estimate cell morphology non-invasively. And I will provide examples from some recent works on how diffusion spectroscopy can be used to estimate the complexity of the dendritic tree, the caliber of the brain cell projections, as well as general astrocytic hypertrophy. And I will conclude with summarizing the take-home messages. To have also another perspective on these topics, I invite you to watch the other educational lecture from Clémence Lignol, program number E974. So why should we use diffusion spectroscopy for cell morphology quantification? So the ultimate goal we want to achieve is the estimation of the brain tissue microstructure. So towards this goal, we could use water diffusion-based MRI techniques such as DTI or NODI that have been shown to be uniquely sensitive to brain tissue microstructure. However, there is a problem. Water is not the best probe molecule that we can use to investigate brain microstructure. In fact, water molecules, although abundant, are ubiquitous in biological tissues. They are in both intra and extracellular space, they are within all the cell types, and they can cross the cell membrane, thus exchanging between different tissue compartments. As a consequence, metrics based on water diffusion, such as the water apparent diffusion coefficient, cannot distinguish, for example, between neuronal atrophy or glial reactivity. In fact, the former would result, for instance, in a reduction of the ADC of the water molecules inside the neurons, while the latter an increase for those inside the astrocytes. The cumulative change in ADC that we may measure may not be sufficient to detect any changes occurring in the tissue unless there is a strong imbalance between the two cell populations. For example, a lot of neurons die. In contrast, some brain metabolites are purely intracellular and cell-specific, in the sense that they are primarily confined within specific cell populations. For example, several studies have reported myoinositol concentrations in astrocytes 3 times higher than in neurons, and choline 6 times higher in glia than in neurons. And also NAA and glutamate there is a large consensus about their confinement in neurons. Therefore, these molecules can be used as cell-specific markers, and their diffusion can be measured using diffusion spectroscopy to obtain quantitative information about cell morphology, intracellular viscosity and tortuosity, and much more. So coming back to my previous example, using now, for example, measurements of NAA and inositol ADC, we should be able to disentangle neuronal atrophy from glial reactivity. Well, this is a massive achievement and an exciting perspective, but can we really do that? Well, in the next slides, I will show you that the answer to this question is a big yes. So coming back, so first of all, a few studies have provided evidences that brain metabolites are mostly free to diffuse within the intracellular space rather than being confined within subcellular structures such as organelles. So this suggests that we can indeed use brain metabolites diffusion to probe the cell morphology. But how? Well, by using diffusion-weighted spectroscopy and properly designed acquisition. So here, let's see how to tune our experiment to measure different aspects of the cell morphology. In diffusion-weighted magnetic resonance in general, we use controlled magnetic field inhomogeneities that we call gradients to measure molecular diffusion. For example, we perturb the homogeneous magnetic field B within the investigated tissue by applying a linear gradient along one specific direction, in this example, X. This induces a controlled spatial variation of the local magnetic field B, which is now a function of the location X in the tissue, in the example here on the right. This is effectively like placing a grid in our system that can be used to measure molecular displacement in the tissue by measuring the differences in the local magnetic field experienced by our probe molecules, either water or metabolites. For example, if a molecule moves from a point A to a point B in a given time with a gradient, we are able to measure a significant difference between local field in B and A, and from that to infer the amount of displacement that the molecule performed in the observed time window that we call diffusion time. Instead, without a gradient, a molecule that starts in A and ends in B wouldn't experience any difference in the local magnetic field, and therefore we wouldn't be able to link this difference to the displacement of the molecule. So in diffusion MR, we use specific magnetic field gradient pulses to spatially label probe molecules and then follow them during their diffusion process. When designing our experiment, it is good to keep in mind that we can control some of these parameters. We can control the diffusion time, as well as we can control the characteristics of this grid by controlling the gradient strength. So a gradient strength equal to 0 would place a grid which is useless, but increasing the gradient strength, we can increase the resolution of our spatial grid. And this means we are more sensitive to small molecules displacements. So a finer grid can be achieved using a strong diffusion gradient, and this enables us to measure smaller displacements. Weak diffusion gradients and long diffusion time are instead useful to measure large molecules displacement. Now, for example, if we want to measure the diameter of a cell fiber, we can use a relatively short diffusion time. But if we want to probe the length of the fiber, we have to use a diffusion time long enough to allow the molecule to experience the distance, the displacement L, equal to the length of the fiber. But diffusion time alone may not be enough. In fact, if we want to be able to measure the fiber diameter, we also need a grid fine enough that allows us to measure displacements as small as the diameter. So we also need strong gradients to effectively be sensitive to the displacement of the molecules restricted in the fiber. Now, this simple sketch, using this simple sketch and keeping it in our mind, we can already suggest general guidelines on how to selectively probe specific features of the cellular morphology. So, for example, if we are interested at the long-range cell morphology, such as cell projection segment lengths and branching, we should use long enough diffusion time and weak enough gradient strength in order to gain sensitivity to these features and lose sensitivity to other features we are not interested, such as segment and soma sites. On the other side, the opposite logic of course applies. So for the case where we want to measure instead the fiber sides and the soma sides, we will need shorter diffusion time and much stronger diffusion gradients. So once we decided our experimental strategy, we also needed to decide which sequence to use for these measurements. In fact, many sequences are available for diffusion-weighted experiments, for example, stimulated echo-based sequences or spin echo-based sequences. The choice of the sequence depends on many factors, including the SNR and the bias due to mechanisms other than diffusion, such as relaxation. For example, steam-based sequences are best for long diffusion time acquisition because they enable shorter echo time than spin echo-based ones. But the SNR, we have to remember, the SNR of a stimulated echo is half that of a spin echo. Concerning which acquisition to use for which measurement, I refer you to one of our recent review for further details. For now, I will just use as general guidelines steam-based sequence for long diffusion time experiments and spin echo-based sequence for medium to short diffusion time experiments. Okay, perfect. So we chose our strategy to achieve the highest sensitivity to the feature of interest, and we chose the best sequence to get decent data. But we did not define actually what long diffusion time with gradient or strong gradient short diffusion time mean. It can mean seconds, hundreds of milliseconds, gradient strength 10 millitesla per meter, 20, 200. So we need to give a little bit of an order of magnitude for these parameters. So perfect. To find the answer to this question, we can proceed in different ways. A fancy one I would like to show now is to use advanced numerical simulations. So here is an example of using our generative model to create realistic brain cells, such as Purkinje cell, tripolar neuron, or stellate neuron. And then perform Monte Carlo simulations of diffusion within these ultra-realistic structures. And we can clearly see here that if we measure the ADC at different diffusion time, these three very different cell morphology have a very different impact on the measured ADC. So what we can also see is that the difference between the different ADC increases and becomes very important if the diffusion time is around 100 milliseconds or above. So we can use this kind of numerical simulations to help us identifying the relevant microstructure features and the best sequence parameters to measure them. So doing so, we can now place numbers to long and short diffusion time, weak and strong gradients. To be more sensitive to long-range features, diffusion times longer than 100 milliseconds and p-value as small as 5 milliseconds per square micrometer is a good start. For short-range features, diffusion time as short as 80 milliseconds and p-value as much higher and p-value much higher than 1 millisecond per square micrometer are recommended. So these numbers hold for metabolite diffusion, which is much slower than water diffusion. If you want to make the same kind of rationale for water, you have to scale this number by a factor roughly 4. So as first example now of application of all this machinery, I would like to show you how we have used this approach to measure the complexity of the cell morphology using metabolite diffusion spectroscopy. So in this work we published a few years ago now, we proposed to use a computational model of complex cellular structure to simulate the ADC time dependence according to morphometric statistics. We can use basically here the curve is the simulation and the data points, the points are the measured data. We can use an iterative optimization to change the morphometric statistics, generate new cell, new simulation, until our simulation fits well the data, and we can in this way estimate the features of the cell morphology, such as the segment length and branching order, non-invasively using the metabolites. We measure the ADC time dependence of 5 brain metabolites up to 2 seconds in vivo in mice and macaque, and the estimated cell morphology from the method I just described to you. As you can see here, we found that cellular morphology for those metabolites supposed to be confined in glia were compatible with the literature. In particular, we measured a factor 2.5 in total glial size between mice and macaque. While for neuronal morphologies, we overall measured estimated similar features between the two species. Then we can use our virtual cellular models to create virtual slice of histology and compare directly with confocal microscopy images from real histology on the same mouse, as you can see here. Our histological analysis and shoal analysis showed that the estimated cell morphology closely was very well matched with the actual cell morphology measured from histology, suggesting that we can indeed access these kind of features non-invasively using metabolite diffusion spectroscopy. But we can do more. In fact, we can focus now on the short-range morphology and measure, for example, the average size of the cell fiber. So in this work, we measure the diffusion of the same 5 metabolites, but at shorter diffusion time of about 60 milliseconds and ultra-high b-value, up to 60 milliseconds per squared micrometer. To estimate the cell fiber diameter, we use a simple geometrical model of randomly oriented cylinders with a given radius. We showed that this model perfectly explained the measured B dependence of the signal and provided very reasonable estimates for the cell fiber radius, with glia metabolites reporting larger radius than neuronal ones. These results have been later confirmed by Shemesh and co-workers and by Vincent and co-workers using a different approach based on double diffusion encoding. However, Vincent et al. Also showed that other features such as soma and branching may indeed affect the double diffusion encoding signal modulation, and should be taken into account when modeling this kind of data. Finally, very recently, experiments performed by Lundell and co-workers suggest that the SNR in human experiments may not be good enough to allow a reliable estimation of the fiber size. So if we want to translate this approach in vivo into human, some more work for optimizing the sequence and the acquisition is needed. So finally, we can do probably what most of you may be thinking by now. Well, let's just join these two kind of measurements and quantify together at the same time both long and short range morphology so that we can characterize brain cells all together. We have recently done it in a proof of concept experiment to probe astrocyte reactivity, a process which involves astrocyte hypertrophy during injury or inflammation. So what we did, we used a mouse model called CNTF, which induced a large astrocyte reactivity in the area surrounding the injection site. We had a 10 mice control group injected with lenti-lac-Z in the striatum, and an equivalent CNTF group injected with lenti-CNTF in the same area. We measured the shorter diffusion time ultra-high B-value signal for 6 metabolites, and we found significant difference only in the myoinositol signal. We also performed low B-value and ultra-long diffusion time experiments to probe the long-range morphology, and again we found significant differences only in the inositol ADC time dependence. These results alone, without any modeling, already suggest that the inositol could be a specific marker of astrocyte reactivity. But we went one step further and we modeled and analyzed the data as I described in the previous slides to extract the quantitative information from both short and long range morphological features. So from the short diffusion time and ultra-high B-value, we measured astrocytes, fiber and soma radius in perfect agreement with direct histological evaluations we performed on 3D reconstructed astrocytes from confocal microscopy from the same mice, post-mortem of course. The analysis of the long diffusion time data also provided the estimates of cell projection segment length and branch order, again in very good agreement with histological estimates. So we overall measured an increase in the soma size and fiber size, as well as an increase in the fiber length, but a non-statistically significant decrease in the branching order. So here is a simple visual comparison between the predicted cell morphology from in vivo diffusion spectroscopy and those measured ex vivo from the 3D confocal microscopy. So of course, ex vivo measurements may be affected by other factors such as tissue shrinkage, while our estimates are of course based on models, so they rely on the model assumption. So there is some difference between the two morphology we estimated, but overall, as we have seen in the previous slides, the numbers of the average measurements are in very good agreement. So to conclude, I hope I convinced you that diffusion spectroscopy provides a unique microstructural information complementary to those provided by diffusion magnetic resonance imaging. If you want to detect neuron-specific microstructural alterations, we can use mostly N-acetyl aspartate and glutamate as molecules which are more specific to this cell population. While to detect glial-specific microstructural alterations, inositol and choline seems to be more specific, although literature is still controversial and more and more studies are coming out supporting this, this assumption. Finally, we didn't comment much about creatine, but because in general creatine is heavily involved in the energy metabolism, and so modeling and interpreting the diffusion of creatine is way more complicated than the other metabolites, but creatine itself can be used as a marker of energy metabolism. However, keep in mind that experimental design is crucial, as I hope I demonstrated to you, and advanced numerical simulations can help to identify the relevant microstructural features and optimize the acquisition for their estimation. That is not the only way to do so. The important take-home message here is the experiment and the way you design the experiments is fundamental. You cannot measure long-range cell morphology if you use too short diffusion time, for example. And finally, also data analysis is very challenging. It requires complex computational tools, appropriate biophysical modeling, and histological reference and comparison with histology is at the moment always necessary to validate our measurements. And I would like to also add that also the spectra processing is very challenging, and the quantification of the spectra is very important, as you may see in discussed in the previous lectures in this session. So I would lastly share with you a list of recent reviews and researchers involved in diffusion spectroscopy that are more than happy to help you and assist you setting up and performing your first diffusion experiments. Feel free to contact them according to their expertise that you can see here and the location. With this, I thank you all very much for watching. I hope you found this lecture interesting and informative. And please email me or follow me on Twitter if you want and if you need. Thank you.